So now from genes to genome. So we now know what the genome looks like, right? The genome is a culmination of all of the genes. Recall that all of the genes can be arranged in a single chromosome. Now this cross-sectional image is showing you if we were to zoom in to a chromosome, let's say chromosome 1, right? So if we were to zoom into a chromosome, we would observe that we see the genes themselves lined up in a similar manner. The region that is in between the genes is what's known as an intergenic region, right? So in between here, you see gene 1, gene 2, and there's intergenic region, and so on until you get to the end of that chromosome. The information we can gather from each of these regions is informative because we have what's known as coordinates. And what you're going to notice is that for any given gene that you look at in the human genome, there are certain coordinates. The coordinates are helpful because that tells you where exactly, what chromosome and what position on that chromosome it's located. So that's really helpful because now you don't have to go around looking for epidermal growth factor receptor gene. You know exactly where it's located. And so if you're looking at the large scale genome viewer, something like what we'll be looking at shortly, you can see where it's located and you can see what's near it, the genes that are near that particular gene. And you have the specific location information. So it's sort of a GPS tracker, right? So really it's a GPS tracker for all of the genes so that we never have to kind of go around fishing for where to find things. So the genome contains genes. The regions, the other regions that aren't included in here, similar to what you see down here, are regions that are what's known as coding regions, which would just imply that's where the gene is located. And then notice here, non-coding region, right? That's the premise, is that it's a non-coding. But again, this information is changing in real time because we are uncovering some role and function for these non-coding regions. Turns out they may actually be doing something that we have yet to discover. Now, finding genes. What example would we want to think about when we say finding genes? When it says finding genes, this is really thinking more about a diagnostic, right? This is really thinking more, you have a sample and you're curious to explore that sample to determine, what might be of interest biologically. Let's say it's a sample you collected from someone that has lung disease or something like that, right? So you would design a simple study where you say, okay, I have 3 normal individuals, no heart disease, and I have 3 that have heart disease. I'm interested in exploring, are there gene differences that could be causing this? Are there some genes that are more expressed and that could be causing the disease? Right, so early questions that were being posed were about genes and pathways. And how we did those in the lab was we just used an animal model, usually a mouse model, or sometimes primates. If they were doing things like vaccine studies, they'll generally use larger animals because those tend to give a little bit better idea of how humans will respond, right, before you test things in humans. Using a primate model to test out a vaccine might actually help because then you can decide the dosing. But for some studies, they may use mouse, or they may use rats, or even for some of the work I did years back, we would use pigs, because their lungs function very similarly to humans, right? So finding genes was really a way of trying to understand how diseases or how phenotypes were inherited. And really the interest there is human disease. It's always the overlying, overarching interest is really trying to diagnose and better understand human disease. So microarrays came on board probably right about 2000, probably about 2008, 2007, thereabout. And microarrays were the first attempt at taking a given sample and using chemistry, really excellent chemistry and physics, because the optics behind this chip are amazing. Now, this chip that you see there, I'm wondering if I have something that's probably big enough to show you. But if you took my phone and slurped it right about there, that's how large these chips are. So they're not massive, right? They're pretty small, but within each of these chips, you can insert a biological sample and place the chip into an instrument that basically does very excellent chemistry protocols and does things like hybridization, incubation, and basically formulates this whole method that tells you the expression level of every known human gene based on pre-existing information that it has on what is expected. So it covers a great deal of physics because it's using light intensity and fluorescence emittance. And the optics is all physics. It's all pure, beautiful physics. And the chip itself is designed using chemistry technology. But this was really our first wave of being able to rapidly assess within, it used to take me about 5 or 6 hours when I did them decades ago. But it would take about 5-6 hours to run the sample and then get your result. But with these technologies, you could easily tell the expression levels of every known gene in the human genome. Now, that information could be very useful. We were able to find things that were overexpressed, right? So then all of a sudden, now you hear the terminology overexpressed, underexpressed, downregulated, upregulated. That would not have been possible without having microarray technology. Okay, so it really is sort of like it was the foundation of what we're now doing with high throughput sequencing, next gen sequencing, right? We say next gen because microarrays were they were first on ground. Now, why is it important to track the presence of genes across genomes? So we can ask a lot of different questions. So for example, common features of different organisms. So sometimes people think about common features, look at things like genomic elements, right? Elements within the genome, and then also variants, right? Genetic differences from human to human, right? And this is part of, my huge interest in ARDS is because it's interesting that there are variants that predispose people to getting ARDS and also not only getting it, but then getting really sick with ARDS. So it's important to track these because of each of those contexts. So databases are all linked to HGP, as we've said earlier, and the types of sequence data available are all here. And I'm gonna pause here because I want us to, this is kind of a good pausing point, but I'd like for each of you all to go into this ENCODE project and let's just spend a few minutes kind of exploring it.